morning. I think everybody has been anxiously waiting for the start of the grand rounds. And so here we are, starting with the first of the season. And I'm truly delighted to introduce today's speaker, Dr. Jin Tang, who hails from Emory University. He received his graduate degree from Peking Medical College and Chinese Academy of Sciences in Beijing. After receiving his Ph.D., he worked there for a year or so and then came to the United States, where he did postdoctoral studies for about 6 years at NIH and then at Johns Hopkins University in the Department of Urology, where he studied molecular biology and prostate and cancer research. He started his academic career at University of Virginia as assistant professor, and then a few years later moved to Emory University as associate professor, and was promoted within a few years to the rank of full professor and also as vice chair of research in the Department of Hematology and Oncology, and has a membership also at the Winship Cancer Center. He also holds the Distinguished Award, Cancer Fellow Award from Georgia Cancer Coalition, which is a major honor for individuals working in the state of Georgia. For a number of years, he's been interested in the study of tumor suppressors and has identified a number of them, including ATBF1 and KLF5. And his interest is in studying the mechanism of how these tumor suppressors operate in normal development, and then how are they dysregulated during the pathogenesis of cancer. So it's with great pleasure that he's going to talk, that I introduce him to you, and then he's going to talk about KLF5, which is his major interest right now.
Well, thank you very much, Khalil. Thank you for having me here. It's a great pleasure to be here and to be the first speaker for this year's grand rounds. As Khalil mentioned, when I started my lab in '96 at UVA, my focus was to identify cancer genes, particularly tumor suppressor genes, and then later years, and I focused on two of them. Group-like factor 5 is one of them, and trying to understand how this gene works in normal epithelial homeostasis, and also when it's inactivated, and how its inactivation contributes to tumorigenesis. I have to say this: I have no financial interest to disclose. My talk today will be in four parts. First, about how I came to study KLF5, how we discovered it as a candidate tumor suppressor gene, and then continued the functional studies to confirm the tumor suppressor, and then how the functions of KLF5 appears to be context dependent. Can be suppressor, can be also a tumor action promoter. And then try to understand how the same molecule can be both a promoter and a suppressor of tumorigenesis. And final part is going to be some what we know about KLF5 and see whether we how we can try to develop something out of the knowledge we learned about KLF5. When I started my lab that time, I focus on chromosome deletion. Chromosome deletion can be in one of the two copies, or can be in both copies of a chromosome. And it's very frequent. It's probably the most frequent genetic alteration in any cancer cells. I mean, cancer cells have mutations, but chromosome deletion, you can always detect chromosome deletions. And even in normal cells, when they passage enough, with the high resolution, like array-based technology, array CGH kind of technology, you can always detect chromosome copy number changes. and it can be induced by carcinogens and can occur in early stages of cell-provision tumorigenesis, and of course there are quite a number of genes now. There are knockout, the equivalent to chromosome deletion, and can induce tumor in different organs, in different model systems. So it is much better understood now about the chromosome deletion, tumor suppression, and tumorigenesis. In prostate cancer, this is a summary of the chromosome deletions based on the traditional CGH studies using metaphysical chromosomes, comparing tumor DNA versus normal DNA. And you can see the most commonly deleted chromosome regions were 8p and 13q. And 10q is where the P10 gene was identified. Actually, the frequency deletion is not as high as AP and 13q. 13q deletion is actually also the second most frequent in the chromosome region among 70 type of different tumors, not just the prostate, like breast cancers and even some like lymphomas or other cells. Many of the regions now have clear candidates of the tumor suppressor gene, like the 10q well-known P10 gene, and AP have this gene called NKX3.1. It is now occurring quite many papers published confirming it indeed has a tumor suppressor role. And my lab worked on different chromosomes, and we identified candidates for 6q. So U50 is a non-coding small nuclear RNA, and also ATBFF from 16q, and FOXO1A from 13q14 is another region of 13q. So there are two distinct regions, deletion and 13, and KLF5 from 13q to 1. So give a little information about how we identify candidates for the chromosome regions. Basically, most important approach was the deletion mapping. We analyzed human tumor samples. I was in the Department of Pathology in Virginia, and Henry Fries, a new pathologist, my collaborator, a strong supporter, and we collaborated, and he got a large number, hundreds actually. He processed hundreds of tissue specimens, and using microdissection, so we isolate DNA and analyze for chromosome deletion and gene mutations. And then expression studies and some in vitro functional studies, and the first stage. And then of course there are other studies we use to identify genes. So chromosome deletion, the basic principle is you analyze different tumors and see the deletion region for individual chromosome. And each line here represents a deletion, the size of that deletion in a cancer sample. So you align them and then look for the smallest region of overlap for that region, and that suggests the location of the tumor suppressor gene. And it works well in many cases, but not always. So using this approach, I mentioned that we found several genes using the same kind of combination of different approaches. And for KLF5, this is a deletion summary of the several key samples and narrow the region to the KLF5 locus. We have several cases here, and the smallest region of deletion was identified in this prostate cancer cell lines, small cell carcinoma cell line from prostate, and that was only about 142 kilobases, small enough to only include 2 genes. KLF5 was the only gene, complete gene within that region. So the deletion mapping gives us good evidence suggesting KLF5 might be more possible candidate for the 13q21 deletion in human cancer. And this is using FISH to verify to see this deletion in LMP one of the prostate cancer cell lines, and only one chromosome has KLF5 gene. But GC3, the other prostate cancer cell line, commonly used, has two signals, so they're confirming the deletion. And so the deletion was identified as the because the deletion frequency occurs like that was like over 40%, so it's quite frequent. And later, the deletion was confirmed in mouse models. You knock out one allele of KLF5, you can get a phenotype, and that means KLF5 is haploinsufficient. It needs both copies to make a sufficient product to function in normal environment. So, but in our study later we found actually not just the chromosome deletion inactivates KLF5. We answered another mechanism. We found it's excess protein degradation, which means KLF5 is regulated by this so-called ubiquitin proteasome pathway, or UPP, which is that degrades protein as a normal mechanism of maintaining a proper level of protein inside cells. During that study, we found that in the UPP system there are three enzymes: E1, E2, E3. E3 is the ubiquitin ligase that transfers ubiquitins to proteins. And once the protein is attacked with the ubiquitin chain, then the proteasome can degrade protein. So we identified WWP1. This is a non-ubiquitin E3 ligase as a target. As a E3 ligase for KLF5. And the interesting thing was UPP. This E3 ligase WWP1 is located human chromosome 8q. That's one of the regions frequently amplified in human tumors, including prostate and breast cancers. And we found WLP1 is also overexpressed, not just so the amplification, of course, the direct consequence of the overexpression of a gene. So UPB we indeed found is overexpressed in cancer samples, and we have also the biochemical and functional studies confirming amplification of WLP1 causes overexpression, and overexpression then leads to excess degradation of KLF5. In other words, in addition to chromosome deletion via haploinsufficiency inactivates KLF5, we have this EQ2 amplification leads to WLP1 overexpression that causes excess protein degradation as another mechanism of inactivating KLF5 in human tumors. So if we combine the two mechanisms together, inactivation of KLF5 in human tumors will become more than 42%. The exact frequency we don't know, but I estimate it could be more than 50% or even 60%. So that's how we came to KLF5 through biochemical, genetic, and functional studies indicating KLF5 could be a tumor suppressor in prostate cancer. And well, then, of course, we studied the function KLF5, and so the first of me as a tumor suppressor, we test some functions. There are functions some in vitro system. This is one of the prostate cancer cell line D145 is in vitro. You get KLF5 expressed in the cells and versus a control, and we see a significant reduction in the cells that could survive or continue to proliferate. So it's inhibition of cell proliferation, or excess cell death that is supporting the tumor suppressor function of KLF5. This paper from a Japanese group, and they tested the KLF5 function in prostate cancer cell lines using xenograft models. So in two cell lines, DU145 and the PC3, they found KLF5 expression can inhibit tumorigenesis in xenograft models. And they found a unique mechanism of ER beta was responsible using KLF5 to in this setting to work as a tumor suppressor. But this study needs to provide evidence in vivo model, using in vivo model, so in tumorigenesis model indicating KLF5 can indeed inhibit tumorigenesis in prostate cancer cell line. And in our, we studied the knockout of KLF5. We generated a mouse model that has this floxed KLF5 allele. So when the allele is floxed, then you can use the Cree recombinase to delete KLF5 specifically in the tissue where Cree is expressed. In our case, we combined with the probation promoter induced the Cree expression model, and that is the probation specific to the prostate. So in this case, we can express Cree specifically in the prostate, then knock out KLF5 specifically in the prostate, only in the epithelial cells of prostate. So when we knock out KLF5 in the prostate, just knock out KLF5 alone, we did not see any cancer phenotype, not even pain or dysplasia kind of changes. We noticed that when we knock out one allele of KLF5, we saw increased cell proliferation, as indicated by the staining of a Ki67 proliferation marker. And the increase was significantly heterozygous genotype. It means knocking out one KLF5 allele, but the difference was not significant when both KLF5 alleles were knocked out. And also, it caused actually the apoptosis was more visible in homozygous allele, which is different from the cell proliferation. We have explanation for that because I mean, KLF5 deletions probably we knock out both alleles, cells probably die. We know now KLF5 is essential for epithelial proliferation. So if you knock out both alleles, then epithelial cells probably die. And but in any case, knockout KLF5 alone did not cause cancer phenotype in the mouse model. When we combine the knockout KLF5 with knockout P10, now P10 is a well-known tumor suppressor now. You knock out P10, the prostate causes pain and also carcinoma in some strains of mice. And so when we knock out KLF5 and the P10, so-called double knockout, and we see a different outcome. In heterozygous P10 knockout background, means we only knock out one allele of P10 and then test the knockout KLF5, and we see a phenotypic change. So even with one allele of P10 knockout, we see knockout KLF5. So one allele knockout or both KLF5 alleles knockout, we see dysplasia, haploinsufficiency, dysplasia, and also increase the cell proliferation, and it's different from the knockout of KLF5 alone because we see more proliferating cells now when both KLF5 alleles knock out. So in the background of a one P10 allele knockout, we see a more consistent kind of data supporting KLF5's inhibitor of cell proliferation. When we knock out KLF5 in the background of both P10 alleles knockout, in other words, so all mice have both alleles P10 knocked out, and then we test knockout different number of KLF5 alleles. And KLF5, it's been well documented in literature. When both P10 alleles knock out, mice get prostate cancer. And this is the phenotype you can see for P10 knockout. And it's still a differentiated carcinoma. And you see the lumen structure. And when we knock out KLF5, we see a progression. I don't know whether it's a real progression, but we see a difference in it. So the structure of tumor is different now. So knock out one KLF5 allele, we see the structure is more disorganized. When we have both KLF5 alleles knocked out, we see the loss of the lumen structure. Quite a dramatic effect by the KLF5. So Also, when we look at the phosphor AKT, it's oncogenic signaling during tumorigenesis, we see the increased expression, especially in this homologous of KLF5 knockout group. So these in vivo studies clearly indicate knockout KLF5 can be a tumor promoting factor in prostatic tumorigenesis. And in other studies, KLF5 has also been suggested as a tumor promoter. So we just talked about the tumor suppressive function of KLF5. We have evidence, but there are also evidence KLF5 can be promoting tumor, including my own study. We have one publication showing that too. This is a study from Vincent Young's group, and so they found KLF5, if you knock out KLF5, actually limits intestinal tumorigenesis. In other words, they have a model, you knock out KLF5, then the number of tumors actually decreased. Well, the explanation can be different, but that is in some way consistent with the tumor promoting function of KLF5. And they have other studies also supporting KLF5 as a tumor promoter. Vincent was an Emory for many years, and we both work on KLF5. He also worked on KLF4, but we were focusing on different functions. I was focused on tumor suppressive function, and he was focused on the tumor promoter function. And we talked about this, and I said that there must be a mechanism to unite us, and it turned out that now I think we have a better answer in terms of the uniting his studies and our studies. This is from one of my earlier publications. We found in this TSU cell line, which was used as a prostate cancer cell line for many, many years until about 10 years ago, the genotyping study confirmed this actually derived from a T24 bladder cancer cell line. One of the examples, cell lines can be contaminated. So anyway, but even as a bladder cancer cell line, it's a very malignant. T24, some people use that cell line. It's a very malignant cell line, has a RAS mutation. And in that cell line, when we express KLF5, it doesn't have high level KLF5 expression. So when we restore KLF5 expression in this cell line, we see increased tumor growth. This is the tumor weight after excision from skin mice. You can see the tumors, larger tumors for KLF5 group versus the smaller tumors for vector control group. And that is different from the study in prostate cancer cell line D145 and PC3. Clearly, KLF5 suppressed tumorigenesis. So this is also, so I mean, we were confused ourselves. How can this same molecule be promoting tumorigenesis in one cell line but inhibiting tumorigenesis in other situations? So then we spent quite a number of years trying to figure out what the mechanism could be. Today we have much better understanding, but we started with cell culture model using a human epidermal cell line, and our hypothesis after some time, and we came to the hypothesis that TGF-beta could be responsible for this opposite effect of KLF5. It took us a long time to come to that hypothesis, but at least in vitro culture cell lines, we have evidence indicating that. And so this next few slides, I'm going to show you the cell lines we did with for the mechanistic study. Mostly we use this human epidermal cell line called the HACT, which is spontaneously modelized epidermal cell line, and very commonly used in studying the TGF-beta function and the mechanism in the 90s and many years, even today still many labs use that cell line. So the TGF-beta pathway is very well understood in that cell line. So we use that cell line because of the hypothesis of TGF-beta. And the turnout is the acetylation, which is acetylation of KLF5, which is induced by TGF-beta signaling, is the key for KLF5's function in and using cell proliferation, because in the in vitro model we only did the tumor cell proliferation analysis. And our data suggests KLF5 is a cofactor for TGF-beta signal. I'm going to show you a few slides. And this is using the cell line to measure cell proliferation. Now, TGF-beta inhibits cell proliferation in the cell line, as well documented. And we noticed when we, this is the control, so without knockdown of KLF5, you see inhibition of cell proliferation by TGF-beta is the black bar here. But when we knock down KLF5, even subtle level of knockdown, 35% or 50%, we see loss of that inhibitor function for TGF-beta, and even increase. So this increase actually was significant. This is a 50% knockdown, and equivalent to a knockout of one KLF5 allele, and there's an increase. But when we have more KLF5 knocked down, cell proliferation itself is actually compromised, because KLF5, if you need KLF5, I mean cells, epithelial cells need KLF5 to maintain the proliferating state. When we analyzed using TGF-beta target genes, one was P15, the other was MYC oncogene. These are very well established targets of TGF-beta signaling. We found the KLF5 actually is critical for TGF-beta to regulate this gene's expression through the promoter binding. So KLF5 can bind to this gene's promoter, and the binding is induced or is actually altered by TGF-beta signaling. And a large number of biochemical analysis eventually show that the change is because of KLF5 acetylation induced by TGF-beta. This is the one slide shows when you knock down KLF5 and the TGF-beta target genes correspond to the KLF5 expression, and this is a cell line confirming the same thing. Just functionally confirm KLF5 is important mediator of TGF-beta function in these cells. Not just that HACT cell line, but also PC3 or several prostate cancer cell lines. We have similar findings. So this slide shows the when we mutated the acetylation site in KLF5, then the effect of TGF-beta is abolished. This is vector control, and the wild-type KLF5, and this is the acetylation deficient mutant. When you can see the TGF-beta can inhibit cell proliferation here and also here, but when we mutated that lysine, so to prevent acetylation, and TGF-beta lost that function to inhibit cell proliferation. And we have more biochemical analysis to verify this mutation can change the transcriptional complex induced by TGF-beta, and including the TGF-beta factors like SMAD2, 3, and 4, and also the P300 transcriptional factor, verifying them. So the acetylation site is essential for TGF-beta to properly function. This is just another biochemical data. Slides shows the deficiency acetylation alters the transcription of TGF-beta target genes. These are all published, so I'm going to just tell you the point instead of the details of data. And the interruption of acetylation, or deacetylation, let's say, not just abolish TGF-beta function, so proliferation regulation and gene transcription regulation, it actually reverses the function of TGF-beta. This is what the promoter report and say. The promoter binding, KLF5's binding to the promoter, the binding becomes opposite in terms of TGF-beta induced binding when we have a mutation or not mutation. So this wild type KLF5 is the acetylation deficient mutant, and they are binding to the TGF-beta responsive elements in MYC gene. You can see this opposite. TGF-beta induced binding for wild type KLF5, but for acetylation mutant KLF5 actually itself binds regardless of TGF-beta. When you have TGF-beta, it actually inhibits the binding here. So based on those studies, we came out with a model like this to explain how, at least in vitro, in cultured cells, in the context of TGF-beta signaling, how KLF5 functions in opposite ways. Without TGF-beta, we found KLF5 to bind to SMAC, and also on gene promoter, there KLF5 binds to MIS1, MYC, and several other unknown transcription factors. In these settings, KLF5 is not acetylated. It inhibits the P15 cell cycle repressor and induces MYC. I didn't draw MYC here, but for MYC it's the opposite. Induces MYC expression but inhibits P15 expression. When TGF-beta signaling is activated, TGF-beta signaling recruits P300 through SMAD4 to 3, and they acetylate KLF5. After acetylation, acylated KLF5 actually now forms a different complex, transcriptional complex, as P300, as SMADs, and MYC is gone in this situation. And after this change, the consequence changes also. So P15 now becomes induced, MYC now becomes suppressed. So this is in vitro cell lines. Based on our data, we concluded this model indicating how KLF5 can be both proliferative and also anti-proliferative. So also we came to the point KLF5 is a core factor of TGF-beta signaling. It's well known TGF-beta is a tumor suppressor, is also a tumor promoter. So at least we know at this stage KLF5 could be necessary for TGF-beta to function as a suppressor. So KLF5 as a core factor of TGF-beta interact with the SMADs and other factors to function as a TGF-beta. So in that setting, inactivation of KLF5, either by chromosome deletion or AQ application, you get insufficient KLF5, so that impairs the function of TGF-beta. So that in the tumor suppressor side of TGF-beta, we can explain how KLF5 could be suppressed also. So, but on the oncogenic function of KLF5, which is probably more useful in terms of translational study, making things as either therapeutic targets or diagnostic biomarkers or whatever, might be more interesting. So in the last section, I'm going to talk about this area of study. So we cracked out the expression of KLF5. Also, it's acidic form versus an unacidic form in normal and cancer tissues of the prostate. And this is a mouse normal prostate. The three different antibodies, this one detects all KLF5s, total KLF5. And can see KLF5 expressions in most epithelial cells and strong staining in basal cells and also some luminal cells. For acidic KLF5s, the staining is only in luminal cells. But for unacidic KLF5, staining is limited to basal cells, which suggests that unacidic KLF5, consistent with the in vitro find, unacidic KLF5 might be more related to cell proliferation, while KLF5 acidic forms might be more relevant to inhibition of proliferation. In castrated mice, KLF5 expression actually changes. This is the intact mice, prostates, and this is the prostates from castrated mice. You see more cells now become positive for KLF5 expression. Now, after castration, the basal cell population increases and luminal cell population decreases. And we believe that increases because the base have more basal cells, so then more cells are become positive for KLF5. When we analyze acylated versus unacylated KLF5, we see after castration, we see more cells become unacylated, have unacylated KLF5 expression, and less for acylated KLF5. These are several human prostate cancer samples staining for different forms of KLF5. Normal human prostate is similar to what we see in mouse prostates. Basal cells have more unacylated KLF5, and the luminal cells mainly have the acylated KLF5. But in cancers, we see a trend of losing acylated KLF5 in tumors and increased expression of unacylated KLF5 in prostate cancer. We don't have, haven't got enough number of cases to do any clinical pathological correlation analysis, but we see this trend. We had actually a large number of breast cancer samples which we also analyzed expression KLF5 in the different forms of KLF5, and we saw some correlation here with the ER status and HER2 status, and and even lymph node metastases. We don't know the exact implication of these findings, but this indicates the relevance of the KLF5 and its acetylation status to human tumors. We are still working in this area to understand more. And we also found the acetylation of KLF5 actually can be modulated by oncogenic signal. For this part, we did some in vitro cell line studies express RAS oncogene in this HECT cell line, and it was interesting to turn out the RAS itself, if we only express RAS, it can actually increase KLF5 acetylation. Or TGF-beta, of course, can induce KLF5 acetylation, but when we have both TGF-beta and RAS, and then the acetylation is decreased. So, in other words, we have TGF-beta, the TGF-beta induced acetylation when we add RAS, and then acetylation is decreased by RAS. And we have more biochemical analysis showing that because RAS oncogenic signal through MAP kinase can phosphorylate SMAD2, SMAD3, and these are the mediators of a TGF-beta signaling in their so-called linker region. And TGF-beta causes the phosphorylation of SMAD23 in their C-terminal region. And the linker region phosphorylation is more dominant. So when RAS is present, the activation MAP kinase causes the linker region phosphorylation, and then even C-terminal phosphorylation is still there, but this linker region phosphorylation actually interrupts the protein interaction between KLF5 and other TGF-beta cofactors. I'm not going through the details of data. I'd like to just come to this model to tell what we believe at this time, how oncogenic signal can interrupt acetylation of KLF5 in the context of TGF-beta signaling. So in normal epithelial cells, you get TGF-beta, then the phosphorylation in the C-terminal region of SMAD2 and 3, and then that phosphorylation leads to the nuclear localization of SMAD, and they form this complex so-called, so let's say the suppressor complex inducing P15 inhibiting make to as overall outcome to the suppression of cell proliferation and tumorigenesis. When we have oncogenic signal, actually recently we have more data using the P10 PR3 kinase signaling pathway. We got a similar finding. Oncogenic signaling, the phosphorylation of SMAD2/3 in the linker region, that phosphorylation interrupts the formation of this normal complex. Only changes it to a different complex, KLF5 make with, and this complex now actually turn to the opposite, inhibiting P15 expression but inducing MYC expression. So if we think about in tumors, activation of MAP kinase or PR3 kinase pathways are quite common, and that if KLF5 is still intact, not deleted, and those pathways, activation oncogenic pathways, can prevent acetylation of KLF5, thus interrupt the function, the suppressive function of KLF5. And more significantly, it's not just the interruption of KLF5 normal suppressive function, it actually makes KLF5 more tumorigenic if the acetylation is interrupted. We recently got some in vivo mouse data showing that. So in two prostate cancer cell lines, PC3D145, we expressed no wild-type KLF5 or this acetylation deficient mutant KLF5 and analyzed tumorigenesis in nude mice. And we see wild-type KLF5 suppressed tumorigenesis. This is consistent with that science signaling paper mentioned earlier. But this KR mutant, this mutant is deficient in KLF5 acetylation. You can see actually larger tumors, faster growing tumors in both cell lines. And this was, we didn't expect, I mean, we thought it might be a possibility, but we didn't expect to see this significant promotion. So in other words, if acetylation is deficient, KLF5 becomes a tumor promoter. Not just in vivo cell line, we have in vitro cell line data, but this is the in vivo tumorigenesis model showing the same thing. Yeah, and because of the TGF-beta connection, so we tested if we block TGF-beta signaling using this TGF-beta inhibitor called SP. It's a chemical. It can inhibit the function of TGF-beta. So it's still in nude mice. In wild-type KLF5, you can see this blocking TGF-beta signaling. You can see the promotion of tumorigenesis, larger tumors for wild-type. But this mutant, because the tumor already grows faster, and we don't see a difference. Somehow in the vector control, this compound actually slows the tumorigenesis a little bit. We don't know whether it's a toxicity or some other reason, because the cell line itself expresses little KLF5. So something probably irrelevant to the function of KLF5 of vector control. But this is also, I mean, this piece of data, we just got it recently, indicates if you block TGF-beta, even you have wild-type KLF5, you block TGF-beta because the cancer cell line already have activated oncogenic signaling, still it can make a KLF5 promoting tumorigenesis. So the unacylated KLF5, I mean, both forms of KLF5 can induce or regulate many genes, and we're thinking probably for the acetylation deficient KLF5 or the acetylation unacylated KLF5, the genes regulated by unacylated KLF5, we might, based on our findings, we believe it is more the oncogenic form. Then you can probably find more genes than is regulated by, induced by the unacylated KLF5. This is some genes we found in the TSU cell line. TSU has activated ROS mutation, so the KLF5 we expressed in that cell line is unacetylated. So these are representing the function of oncogenic function of KLF5 unacetylated form. And there are many genes regulated by KLF5, including this is different factors. I mean, this is one of the molecules we discovered earlier and later validated that can actually mediate KLF5 tumor promoting function in breast cancer. So overall, this is the model we have now for how KLF5 functions in normal epithelial homeostasis and how its inactivation contributes to inactivation or deacetylation caused by oncogenic signal contributes to tumorigenesis. So in normal prostate, KLF5 is probably unacetylated for the transient amplifying cells, which are proliferative and continue to proliferate and then eventually differentiated to become luminal cells. And when TGF-beta, the differential signal, like TGF-beta is activated, and then these cells probably KLF5 is acetylated in these cells and then induce the cell differentiation. So if KLF5 is deleted via chromosome deletion or excess protein degradation and don't have enough KLF5, and then the differentiation process is probably impaired, and then tumorigenesis could occur. Or if we have other oncogenic signaling is activated, and can also interrupt KLF5 acetylation and make tumor, make cells become tumor. We also believe interruption of KLF5 acetylation could contribute to the progression of the tumorigenesis, but we don't have the evidence at this moment. We're working some in vivo models to see whether KLF5 deficiency in acetylation can promote tumorigenesis, progression and metastasis and that kind of stuff, which is quite possible, but we don't have any data yet. That's what I have, and many people contribute to all the findings I presented today, particularly these fellows and students for the discovery of KLF5, understand the biochemical basis of KLF5, and the knockout mouse studies and the tumorigenesis studies of KLF5. I'll stop here and take your questions.
Questions?
So yes. Which version is targeted by the ubiquitin ligase, acetylated or unacetylated?
That actually we never tested. I would propose to probably the acetylated or both form. I don't know. Yeah, we never tested that. If we think about it, acetylated KLF5 is a relatively small amount of the total KLF5, so it's more likely it's unacetylated KLF5 targeted by the E3 ligase. Yeah.
At what stage of progression do you see loss of KLF5?
Deletion of KLF5 is actually much more common in advanced tumors. Yes, we even actually just analyzed the deletion of 13q. We actually had a publication before. Loss of 13q to 1 this region with that before we knew KLF5. It's associated with the tumor progression in prostate cancer using human tumor specimens.
So in theory, that could be occurring after the oncogenic signal then?
It could, yes.
In the back.
So at first glance, with the immunohistochemical studies, it looked like expression with the total KLF5 antibody was both cytoplasmic and nuclear, and that there might have been, and I don't know if it was just my view from back here, slightly different patterns as far as subcellular distribution of the acetylated and unacetylated forms. I was wondering if you've done any self-fractionation studies to look at that, and if there's any proposed cytoplasmic functions for either form as far as sequestering other transcription factors or such.
We had some data in that fraction that we separate the cytoplasm versus nuclear fraction. And the KLF5 is more abundant in the nucleus, but significantly also expressed in the cytoplasm. And actually we found the binding with SMAD4 is actually without TGF-beta is more mainly detecting the cytoplasm, which is different from the previous view, but SMAD4 only expressed in the cytoplasm with TGF-beta.
I noted that when you looked at the effect of the mutant KLF5 on the growth of PC3 and with DU141, did you look at the androgen receptor containing cell also?
No, we haven't done anything in terms of androgen receptor or androgen. We haven't changed the androgen level.
Also, I was wondering if the WP1, the ubiquitinase pathway, is that increased from during progression of prostate cancer?
That I'm not sure. I don't think anybody specifically addressed that point. The AQ amplifications reported in prostate, breast cancer, different tumors, and in that kind of studies, you just use compare tumor versus normal. Yeah, I don't know. Good question. Yeah. Yes.
So does KLF5, does it alter the metastatic potential of any of these cells depending on how you or in the animals where you alter it?
Well, in the animal studies, we only express wild type versus the mutant. But in human tumors, advanced tumors, metastases definitely have much more frequent deletion of 13q21.
But when you alter it experimentally, have you seen any
Changes
in metastatic potential?
Yeah, we haven't done any metastasis experiment. Yeah, no. Yeah, we actually, I would propose the unAc-Klf5 promotes metastasis if we have a appropriate model to do that. Yeah. Yes.
Do you think proteasome inhibition might be a good therapeutic target for those patients that have high KLF5 degradation? Inhibition of the proteasome so that you're increasing the level of KLF5?
Well, in the WLP1 overexpression cell line, well, like a PC3 prostate cancer cell line has overexpression of WLP1. When we knock down WLP1 in PC3, it increases the KLF5 level, it inhibits cell proliferation. We didn't do the tumorigenesis part, but in vitro knockdown. So in that sense, you can manipulate the WLP1 or the U3, the UPB pathway to restore. Or in other words, let me say, so the like Gleevec, the proteasome pathway, UPP inhibitors, E3 ligase inhibitors, proteasome inhibitors. KLF5 could be one of the factors contributing to the inhibitory effect of those drugs. Yeah.
On the same note, then, do you have any concept of what the deacetylase is that may actually be the deacetylation of KLF5 and then switch the unacetylated form that promotes?
We don't have any data on that.
That would be a nice target.
Yeah. Yeah, that's true. That's a good point. Yes.
Other question? Thank you very much.
Thank you.